think maybe, Maite, that we're just going to start, and then you can let people in as they jump on this train. Welcome to everyone. My name is Øyvind Paasche. I'm the chair of the Scientific Steering Committee of Synoptic Arctic Survey. And as I guess most of you know, the Synoptic Arctic Survey has been going on for a few years. It's a bottom-up initiative trying to coordinate research vessels going into the Arctic and to collaborate and to create a synoptic view. And so it started up in 2015, and now we're looking back at three actually successful years of cruises. So 25 research cruises with the participation of 11 nations, I think is something to be proud of. I think it's a testimony to the importance and value of a science community that pursue progress through collaboration. And it has arguably never been more important than just now. Why? I think in part because the Arctic has now become a politically contested area where we had a good collaborations with the Russian colleagues. We no longer have that, and Russia has also withdrawn from the Arctic Council. So there's, or at least they're not engaged, so to speak. So it poses certain new challenges for the science community, but also obviously for the political sphere. And the other thing I think is probably obvious to everyone attending this meeting is that the Arctic itself is in the process of being fundamentally transformed or renewing itself, if you want to have a more positive outlook on it. SAS is now working to amass results and to make them available, as well as also building on the momentum that we have for another international SAS for 2030, simply called SAS 2030. And today we want to showcase some of the breadth and the width of the science that has come out of these science cruises under the SAS umbrella. And I think we're very lucky to have three presenters today to share results with us, ranging from US, Japan, and Norway. So quite a span. The order, as I am informed, is that Ashley Arroyo is first out, then it's Mariko Hatta, and then third out is Reinholdt. Jensen. So with no further ado, I think I will give the word to Ashley, and also inform you that you can write questions or comments in the chat, I think, and we will open up for questions after each of the individual talks. The escape routes from this meeting is in the back of the room and to the side, in case you have to leave. So the first meeting, the first presentation, Ashley, the stage is yours.
Hey, can everyone see this okay?
Yes, that's perfect.
All right, great. So thank you for having me. I'm Ashley. I'm a PhD student at Yale, and today I'll be talking about some of the results from my PhD so far, mainly focused on dissolved oxygen in the upper Canada Basin water column, its variabilities and drivers over time. And so I'll be pulling on results from these two papers listed here. And I'll mention that the second paper is in review with JGR Oceans, the special collection on the Beaufort Gyre. And I'll also take a minute to mention that that special collection is soliciting papers, and the deadline is not until October at some point this year. And so if you have any questions on that, I'm sure Mary Louise would be very happy to answer those. So I'll just get started by mentioning some of the oxygen observational data sources that I'm using in this study. And so first off, I'm using data CTD profiles from the Beaufort Gyre Observing System Joint Ocean Ice Study annual cruises up to the Canada Basin. And this is aboard the Louis S. St. Laurent. And it's a collaborative effort between Canada and the United States. And so this program consists of annual cruises up to this Canada Basin region. This is an example of the 2023 cruise track. And so here you can see examples of the CTD cast locations. And I'll also point out that these red squares are the locations of 3 moorings that are a part of the program as well. And this is an annual cruise, so it occurs at some time in the fall of each year since 2003. And so we complement those cruise data with data from the ice tethered profilers, which provide a seasonal record of the oxygen. And so here we can see a map of the region, and we have the ITB drift tracks colored by time to show the time span of the observations we're using. And then on the right here, we have a schematic of the ice tethered profiler, just to see how that works. And so now we can start by getting an idea of what the upper Canada Basin water column looks like and how that varies seasonally. And so these profiles are from ice tether profilers. The blue profile shows a winter profile and the orange shows the summer. So we can look at representative profiles for potential temperature, salinity, oxygen, and oxygen saturation. And so looking from the top down, we can see that during the summer, we have mixed layers that are very shallow, fresh, have low oxygen that's slightly undersaturated or around 100% saturation. And so here, this dashed black line is the 100% saturation. And then during the wintertime, we have deeper mixed layers that are saltier, have much higher oxygen concentrations, and are usually super saturated. And so you can see that here. And then, these profiles are both from different ice tether profilers, but both at the same location of the green star. And so now looking below the mixed layer, we have Pacific source layer. So we have the Pacific summer water layer, in addition to the Pacific winter water layer below that, which I'll talk about the Pacific water later in this talk. But now I'll start by discussing dissolved oxygen in the mixed layer. And so dissolved oxygen concentrations in the mixed layer are tightly linked to the sea ice concentration. And so here were the dots, the colored dots are mixed layer oxygen concentrations. And these are the hydrographic cruise data from the Beaufort Gyre Observing System Joint Ocean Ice Study cruises. And so below that, we have the sea ice concentration at the midpoint of each cruise underlying. And so you can see that under high sea ice concentration, you tend to have higher oxygen concentrations. And then you can see this clear transition across the sea ice edge to lower oxygen concentrations in open water. And so now we can further investigate the link between the two by looking at the seasonal cycles of each of these. And so on the top, we have sea ice thickness and sea ice concentration. Thickness is in the black on the left, and these thickness data are derived from those 3 moorings that I mentioned. And then so the sea ice concentration is here on the right in blue. And then these bottom 2 panels show the seasonal cycle of the dissolved oxygen in the mixed layer as measured by ice tether profilers. And so here in the middle, we see the seasonal cycle measured by 3 ice tether profilers that provided a particularly long record. And then on the bottom, we have the seasonal cycle of all the ice tether profilers mentioned in this study. So there's 21 of them, all in the gray lines. And then the mean curves in the black line. And so what you can see from these plots is that the seasonal cycle of the oxygen strongly follows that of sea ice. And so there's increases throughout the winter time as ice is growing and there's less atmospheric exchange. And then we see this really sharp drop off. And that's occurs when the sea ice begins to melt. And so to investigate and quantify the exact drivers, we turn to a one-dimensional modeling approach in which we model each of the processes that are influencing oxygen change in the mixed layer using the ice tether profilers. And so here we have expressions to compute the amount of oxygen change we might expect for each of these processes. And for these, we use parameters computed from the ice tether profilers, such as mixed layer depth, density, and oxygen. And then we also complement those with other data sources, including sea ice concentration, atmospheric pressure, and wind data. And so these are computed at a 1-day time step. And then so for other processes that also influence oxygen concentration, we cannot compute in a one-dimensional approach, including biology and lateral advection. We touch upon those a little bit more in the paper about how the potential influence of those in this context. So now we can look at one example of that. So on the left here, we have results and observations from ITP 85. And so on the top, we have a depth versus time plot for oxygen. And so here in the gray, you can see the mixed layer depth. And then in the middle here, we have the sea ice thickness and sea ice concentration. And then on the bottom, we have modeling results that we compare to the observations from the ice tether profiler. So the observations are in the thick black line, and then the modeling results are showed in the shaded region. And so from this, we can tell that the seasonal cycle, as shown from the modeling, is primarily driven by sea ice growth and sea ice melt. So brine rejection during sea ice melt, increasing the oxygen in the mixed layer, and then meltwater dilution during sea ice melt, leading to the sharp decrease. And so you can see that this modeling approach does a pretty good job at capturing the observations as measured by the ITPs in this case. And then here on the right, we have the same thing for the 21 ITPs in this study. So you can see in the red shows the modeling results, and in the black shows the mean curve for all of the 21 ITPs. And so you can see that generally the modeling does a pretty good job at capturing the seasonal cycle as measured from the observations. And so now, to understand how the mixed layer and the oxygen is changing on an interannual time scale, on the top here, we have a time series of mixed layer oxygen over the time frame as measured. And you can see the linear trend is about negative 0.99 plus or minus 0.05 micromoles per kilogram per year. And these dots are colored by the month. And so now we can compute monthly trends of oxygen in the mixed layer. And so on the y-axis, we have the trend in micromole per kilogram per year. And the gray bars are showing the trend in oxygen. So as you can see, from all of these gray bars, we have a decreasing oxygen trend in each month. And then further from there, we can compute the change in oxygen solubility expected. So the change in oxygen you might expect due to changes in water properties. So changes in temperature and salinity. And then so one of the most prominent changes here is that there are these significant and large decreases in oxygens, particularly during the winter months. And in those months, these oxygen changes are not being driven by the changes in solubility. And so here we hypothesize that this is due to there being a looser ice pack with more leads and fractures in it that are allowing for more atmospheric exchange and outgassing of this supersaturated winter mixed layer to the atmosphere. And so now we can switch gears a little bit and move down in the water column and look at the Pacific summer water layer. So here again, some representative profiles of temperature, salinity, oxygen, and now we have chlorophyll. And so these are at this location of the green star, and these are from the cruise data. And so here we define the Pacific summer water layer from of the waters between 30 meters depth and the bottom bounding isohaline of 32.3. And those bounds are shown here in the dashed lines. And so one of the most prominent changes that we've observed is this strong warming. So the blue is showing an earlier profile from 2006, and then the red profile is from 2020. And so you see this really prominent warming of the Pacific summer water layer. And then what we've also observed is this really strong decrease in the oxygen in the same layer. And so that's what we will look to investigate more. And so here we are looking at, in the columns, we're looking at the different year groups, which are labeled at the top. And then the top row is now showing Pacific summer water layer average potential temperature. And then the middle row is showing the layer average oxygen. And then on the bottom, we have the thickness of the layer. And so what we can tell from here is that we have this really strong warming throughout the Pacific summer water layer. That's also accompanied by this really strong oxygen decrease. And then I'll also mention that this layer has been thickening over time. And so as you might expect, there's lots of variation around the boundaries of the gyre in both temperature, oxygen, and thickness. And so for that reason, we decide to focus for the rest of the talk on this central Beaufort Gyre region, which is shown here in this box. And so now to get an idea of how oxygen has evolved in this central Canada Basin region over time in context with the warming, we can see on the top, we have temperature salinity diagrams. And then on the bottom, we have O2 salinity diagrams. And so the Pacific summer water in all of these are bounded by the dashed lines. And then so in these profiles, or these plots on the left, we see all of the profiles from the central Canada Basin region. And then on the right, we have we've computed the mean profile for each year. And so what you can see very clearly is that there's this really strong warming, and these dots are colored by time. So you can see this really strong warming within the Pacific summer water layer, which is also accompanied by this strong oxygen decrease. And so now what we can do using these mean curves is we can compute anomalies to understand exactly where this maximum change is occurring for both these properties. And so again, here in the columns, we have the different year groups. And then on the top, we have those anomalies in respect to the mean profiles for potential temperature. And on the bottom, we have the same for dissolved oxygen. And so again, you can clearly see through time this strong warming of the layer accompanied by this strong decrease in the layer in dissolved oxygen. But it's important to note here that the maximum anomaly in the temperature sits higher in the water column than the maximum anomaly for the oxygen decrease. And so this kind of hints that there are different processes affecting different parts of the layer and driving that oxygen decrease. And so we further investigate that by splitting up the Pacific summer water layer into an upper portion and a lower portion and compute linear trends in those. And so what we find from doing that, and so here we split up the layer into the upper and the lower at the temperature maximum. And so that also tends to coincide with the chlorophyll maximum in the same layer. And so what we can see by breaking up the layer into these two portions is that the decrease in oxygen in the upper layer is fully driven by the change due to solubility. So warmer water can hold less oxygen, and that effect fully drives the change in the upper Pacific summer water layer. And then in the bottom, or the lower Pacific summer water portion, we find that the decrease in oxygen is partially driven by that same effect, so the decreased solubility due to warming, but it's also partially influenced by this increase in AOU. So that relates to increased oxygen consumption. And so now I'll say a couple words on the source waters. And so this ventilation and the warming effect, so this solubility effect. So we look at the source waters of Pacific summer water to understand this mechanism driving oxygen decreases in the basin. And so these source waters are this Chukchi Sea region, which you can see from these plots on the top. This region is warmer, saltier, and tends to have lower oxygen than the surface waters of the Canada Basin. And so the idea here is that these surface waters have been warming due to reduction in sea ice, And so they can hold us oxygen, and those we're essentially seeing that signal in the Pacific summer water at depth in the Canada Basin. And so here, these are 10-meter potential temperature in the black, and then the oxygen is in the red. All data from this region encompassed in white here. And so you can see the inverse relationship clearly between the two, so indicating the solubility effect. So the idea here is that the solubility is due to the warming of the surface waters, and that's the signal that we're seeing at depth in the basin. And then, so quickly, just to summarize, the dissolved oxygen concentrations in the mixed layer are spatially and seasonally linked to the seasonal evolution of sea ice. And then those mixed layer concentrations have been decreasing during the winter time, which suggests increased atmospheric exchange, and that's likely due to changes in the wintertime sea ice pack. And then the Pacific summer water layer that has seen warming has also been accompanied by decreases in oxygen concentration and concentrations in both the upper and lower layers over this time frame. And so that decrease in oxygen is driven by both physical and biological mechanisms. And that's all I have. So thanks for listening to me.
Excellent. Thank you very much for a really nice presentation, Ashley, and super interesting results. I think we'll just open up if people have questions or comments. Let us know. Did you raise your hand, Manuel, or was that just a mistake, or no? Yeah, there you are.
Yes, thank you very much. Thank you, Ashley. Very interesting and beautiful data you have shown us. As I'm a biologist, I was, well, like that you mentioned a little bit of biological influence, but you didn't mention too much about photosynthetic oxygen production. That in my experience also may influence the balance between those two. So do you know anything about the development of that or changes in the primary production in the area?
Yeah, so in terms of the Pacific summer water layer, where we see that high chlorophyll concentration, part of that AOU increase is likely due to increased oxygen consumption, and that could be because of increased primary production at the surface. In terms of the ice tether profiler data, a couple of the ITPs have chlorophylls on them, and I've not seen any increased signal of oxygen based on the chlorophyll, but primary production is really hard to compute, especially with limited parameters. And so we've looked at ways of doing that. But basically, the dominant signals seem to be the physical processes. And so, yeah.
Okay, thank you very much.
Other questions for Ashley? I was curious about these cracks and the fractures in the sea ice you mentioned in terms of enhanced exchange between the upper water masses and the atmosphere. Could you say anything more about, like, to what extent? Is this an increasing trend, or and how efficient is it really?
Yeah, so small cracks and leads and fractures and things like that in the sea ice are really hard to resolve in sea ice concentration data. So what we've done is looked at the sea ice motion from the ice tether profilers, so that can be computed. And we've seen increasing trends in the sea ice motion that indicates a looser and more mobile sea ice pack. And so that's how we're able to hypothesize that there's the looser sea ice pack and more increased atmospheric exchange. But I think that mixed layer becoming more undersaturated and losing oxygen is more evidence of that. But yeah, those small leaks and fractures are really, really hard to resolve.
I would imagine. Okay, then there's like a final chance here to ask Ashley a question or even a comment. Such nice data. Yeah, Shige, please.
Thank you, Ashley. Interesting talk. I have a question. Are you going to extend your analysis for deeper layer, for example, the Pacific winter water and lower alkaline water? Because I am interested in the oxygen concentration change in the deeper layer.
Yeah, so I've actually looked at the Pacific winter water, and there are changes on the same in the same direction, but they're about an order of magnitude less. And so I didn't further look into those mechanisms. I actually haven't looked at the Atlantic water specifically, but as you can one of those figures I showed was of the whole water column, the oxygen salinity and the temperature salinity, and you can see that there are large oxygen decreases in the Atlantic water where there's not associated warming. And so I haven't specifically looked into why that might be, but it would be interesting too.
Okay, thank you.
Great. Okay. Thank you again, Ashley. And you just have to imagine the thunderous applause you're getting now from all the participants. And we'll move on to the second speaker, Mariko Hatta from Jamstack. And you have the title Trace Metal Measurements in the Arctic Ocean. Are you ready?
Yes, I will try.
Yes, excellent. Go ahead.
First, I think it's this first. And wait a sec. What I'm doing? Here we are.
Perfect.
Ta-da! Do you see my first slide? Great.
Yes, we do. Go ahead.
Okay, perfect. All right, thank you very much for the opportunity to give me this time. I'm not speaking that fast as Ashley did, so let me speak a little bit, take time. But anyway, my name is Mariko Hatta. I am Japanese, born in Toyama. Some of the people might know Toyama is, which is unusual for scientists know, but last time ASSW was held in Toyama, which was my hometown. Anyway, I was working as a chemical oceanographer in a geotraces group for many, many years. So at the time, 2015, I was actually in the Arctic Ocean with the Hilly cruise. And it's not really the study focusing on the Arctic, more like a global ocean. I'm going to show you in a second what I have done so far. But today it's like, I'm more likely overview what we know at the moment based on the geotraces cruises, particularly focusing on the trace metal, emphasize why it is important, who cares about those, why, and also what we can do, how to make it to the better observation and how to collaborate with people. Hopefully you are all get some ideas by end of today's talk. So I'm very interested into measuring trace metal, especially iron, aluminum. My advisor / colleague was British, so I taught me about the accent all the time, and also manganese, and also the micronutrient. But it's like to understand about the spatial and temporal resolution of trace metal distribution global wide. I went to many, many different cruises and all over the world. So this is the map I have been so far. To do this, like a high resolution mapping of trace metal using shipboard analytical techniques, I have been using it. But it's like I have been involved to Kleiber or geotraces or couple of this Antarctica research cruises as well. And also, like recently, I participate into the nutrient intercalibration cruises, which is basically comparing about the several different methods and different group of people to how to do this for shipboard analysis and what is uncertainty we have so far. But it's like for focusing on the Arctic, that everybody on this talk probably know already very well for Arctic Ocean. So I'm not gonna go too much detail just for the overview. The significant loss of sea ice was observed over the decade. And this retreat and loss of sea ice affect higher and lower latitude in the Arctic Ocean very differently. And significant difference in light condition likely lead into the increase of biological activity and more importantly, changing in the timing of the bloom, which also differ into the future prediction by biological modeling. They're based on the sea ice area, and there's many different the scenario was predicted. That's because of at the moment is a little bit uncertain for biological process and what is going to be happening in the future. So we are community urgently need to understand the Arctic biogeochemical process and under this rapid change conditions. Oops, it doesn't go. So this is a at the moment, primary productivity in the Arctic Ocean is increasing. This left panel showed that 3 different time series, which is top one was a mean open water area and chlorophyll A, and also net productivity. There is clearly 2 different patterns separated with a different color. The red color is showing open area, which is loss of sea ice, was very strong positive correlation with the net productivity. The indicating is the more light was increasing our productivity in the Arctic Ocean. But the more recent ones is it is a little bit increasing open area, but it's more like corresponding with chlorophyll A concentration. And then it's a light figure show the difference of chlorophyll A over the decade, and there are more positive difference are showing in the red color and showing in the very big shelf area, Barents Sea and Chukchi Sea and Siberian shelves, but some of the area is a negative effect. As you see, is a very different variability and that depend on the way you are studying it at. So it is important to comprehensive approach to based on regional and also overall monitoring across Arctic Ocean. So that's why the Cameron Synoptic Arctic Survey. And I made this plot based on the science plan for my view using a 5W and 1H. But it's like from the beginning, everybody know that is a Synoptic Arctic Survey is a comprehensive research initiative focusing on understanding the current stage and transformation of Arctic Ocean, particularly water mass, marine ecosystem, and carbon cycle, including ocean acidification. But it is a goal to set the baseline for future study and understand how we informed for the future changing. So like in this talk, I wanted to highlight the importance of trace metal. For example, iron, which is the essential micronutrient for phytoplankton. And I will introduce recent trace metal research work from the Geotraces Arctic and hope to encourage collaboration in the future in collecting and analyzing trace metal as a key parameter for monitoring future Arctic research cruises. So the biological pump is a critical component of ocean carbon cycle. This transfer of carbon from the surface to the deeper water, and some of this may buried in the sediment for a long time. But it's mainly operated by biological process, primarily driven by microphytoplankton in the shallow depths. So it's like the level of nutrient is in the surface water play an important role of biological activity. This is the schematics, is how it works, and how it's important for metal is. Phytoplankton needs to take up the carbon, phosphorus, and nitrogen. That's called the micronutrient. But to get to this micronutrient, the cell needs to use metal and metal enzymes. There are, for example, for carbon case, the cell needs transport bicarbonate from the water into the cell and change it into carbon dioxide. This process requires enzyme and some kind of metal like zinc, cobalt, and cadmium. And when, in case of nitrogen, the organic nitrogen requires nickel, and nitrate case is iron. If you're using nitrogen gases, a lot of iron and molybdenate. So without those metals, the phytoplankton cannot grow. So it's like they needed to have some metals to do this process. There are, for example, the iron case is very insoluble in oxygenic water, like modern seawater, and it's easy to scavenge from the water column. There are iron limitation may occur certain area in the world, like they call high nutrient, low chlorophyll region. For example, North Pacific water or Southern Oceans. Currently, Arctic is not iron limited. Well, in fact, everybody knows that might be familiar with nitrogen limitation. But it is important to understand iron distribution and its process, because that might be changed with the climate warm and ice are getting lost. And especially, as I just told you about the nitrogen require iron to adapt to the phytoplankton growth. And the iron concentration is 1,000 times or even more lower than the presence in the water column. So tiny bit of difference will affect for the phytoplankton activities. But as I said, iron or another micronutrient are bioavailable, which means consumed very easily. And then top of this is Arctic is very complicated area, and there are many different sources, potential sources exist for iron. For example, the iron coming from the sediment or aerosol, or sometimes is a hydrothermal vent in the bottom. It actually exists in the Arctic Ocean as well. And some of the water exchange from the Pacific or Atlantic water and transport and consume. So since this complicated mechanism, we were using a different metal. For example, for my favorite metal is aluminum, which is identified with the dust deposition or aerosol input or riverine input, and sometimes shelf input as well, a shelf or like sediment input. And also manganese is a very good indicator from shelf transport. So the tag along with the multiple element together to understand this complicated Arctic Ocean is important. So the summary on this slide is to understand the source of fate of iron or with the supplier. The multiple element approach were important to understand the diverse input passage and associated processes within the Arctic Ocean. That's why like geotraces cruise are conducted 2015. As a part of international geotraces program, US, Germany and Canadian research group coordinated oceanographic research cruises in the different part of the Arctic Ocean. I was on the Healy with the US team to go to the North Pole and is coming back to do this red line cruise line I was participating in. But geotraces, the iron, for example, or any metals, the ship is big chunk of the metal. So this is to avoid any contamination from the boat. We have to have a specific instrument. For example, we have a geotraces trace metal clean rosette system, which is specially made for the non-metal product or coated everything and try not to have any surface exposed. And also collect the sample in the clean air filtered room, which is built like a 20-foot container, modified it to trace metal clean sampling system, a sampling room to do this, collect the samples. And some of the metal are analyzing using a flow injection analysis like I did on this picture, or the other group or the other metals was collected seawater and bring back to the shore-based laboratory and analyze with the ICP-MS. So this is the result I just show you for what I got during the cruise. This is that the cruise was started from Dutch Harbor, but it's from the Chukchi Shelf to the western side of Chukchi borderland to go into the North Pole, and coming back from North Pole through the Canada Basin, and then coming back to the shelf region. Maybe I can just raise a point. And then those are plot showing upper panel is iron plot, and then bottom panel is manganese plot. To the left is the Chukchi Shelf area, to the North Pole around here, and it's coming back through the Canada Basin. The warm color is a higher concentration, and cooler color is a lower value shown. And as you see, is a strong elevated iron and manganese value are observed near the Chukchi Shelf. And then that's also seen in the manganese distribution, which is showing that dissolved iron and manganese share the same source. And then toward to the interestingly in the North Pole area, which is correspond at the which is underneath of thicker sea ice, is also high value of the iron are observed. But we didn't see anything about elevation for manganese value. The interior iron and manganese value are both very low. And then also, especially iron value are unusually lower compared to the rest of the ocean, which indicating is a less vertical transport to the shallow depths to the interior. The Jensen et al. 2020 are focusing on the source and the fate of iron and manganese in the shallow Arctic Ocean. This is the same interior, same data set from the Chukchi Shelf to the North Pole and coming back to Chukchi Shelf. And top panel is a salinity, silicate, dissolved iron, dissolved manganese, particulate iron, particulate manganese are showing. This is both showing dissolved iron and manganese share common source and releasing during the diagenesis in pore water of Chukchi Shelf, but they quickly decouple along the transit. There is a three-part stage of decoupling expected to happen, seems to be happening. The iron released to the oxic shelf water, iron was rapidly oxidized to iron hydroxide and reprecipitated. That will affect more than the manganese. And the shelf break area, dissolved iron is removable more quickly than dissolved manganese. That's because a lot of particle scavenging going on, and seems to be dissolved iron is more absorbed than the manganese onto the surface of particle. Towards the offshore, more than 1,000 kilometers away from the source, there is a less particle abundance, so less particle scavenging, but it's a colloidal formed iron, which is combined with the ligand or another organic compound. Species was stabilized within the water and remained in there, and rest of the form of the iron or manganese is slowly removed from the water column, but it's stay the iron in the water. But anyway, in summary, iron and manganese originate from the common source on the shelf, but undergo towards the offshore, different removal fate and process is occurring in the water column. And now focusing on another interesting feature for the iron underneath of thicker sea ice. This is the surface plot on the cruise track. In the surface water, higher value of the iron was observed in the Central Arctic Ocean. This is also seen the high silicate value and chlorophyll A, but interestingly, correspondingly elevated colored dissolved organic matter, CDOM, are observed, which is suggesting the presence of heterogeneous organic matter. The possible source of iron enrichment could be a two potential factor. One is melting of sea ice on the site. There is a very thick sea ice there. And also advection of the advection of the another water masses in the Central Arctic Ocean. To identify the process of the source, now looking at more detail of the sample under the sea ice at the ice station in the Arctic Ocean. So this is what we did is we get onto the ice floor and making a camp and making a drill and a hole and to put on the trace metal pumps tubing in there and pumping up the water with a trace metal pump system. And also the ship itself has a rosette and trace metal clean rosette to collect the samples. And then ice team wearing a white clothes and trace metal team, sea ice team collecting sea ice itself and snow and melt pond. This is the vertical profile we obtain from the ice stations. The vertical profile was salinity, silicate, and iron. All of the station is at the big ice, so ice stations. Up at 20-meter depth we will collect the sample using a trace metal pump system, and the deeper than 20 meter is from the ship. Because try to not get contamination from the boat, so we are separated two different ways to get to the complete for the profile. And good news from this profile is showing the pumping system, 20-meter depth, and vertical profile from the ship is matched very well, which means the both does not contaminate it randomly. And then from the figure showing the very strong stratification with the low salinity water at the very shallow depths, and then increasing with the depths. And silicate also very low at the shallow depths and increase with the depths. But iron was a two different feature. 3 stations out of 4 was very high value, about 2 to 4 nanomolar. And then one of the stations, which is a very south, close to the Canada Basin, was a very low for iron value. So then we were looking into it, what is the concentration for the sea ice, if it's a sea ice or snow or any other thing affected the water column. But the value of the iron is very low. And even like this is the summertime, it's all of this brine water might be a drawdown, but still concentration is 3 to 5 times higher in the water than the ice. So then, like we looking at the geologically the stations, we did see that higher value of the iron was within the transporter drift, and then very low value we'll find is within the Beaufort Gyre in the Canada Basin. So this is indicating the high iron water was transported by the current underneath of transporter drift on the ice. And then the silicate value is also corresponding in the similar to the iron as well. Then like metric water percentage was calculated using salinity and oxygen isotopes and indicating a coastal influenced water observed in the higher percentage of the coastal water presence in the Central Arctic Ocean. And also the radium isotope value are also elevated in the same water mass. The radium isotope are continuously produced through the radioactive decay of tritium and uranium in the sediment. The radium isotope is a very good tool to identify the interaction between seawater and coastal sediment. And then the Kip et al. 2023, they find it is the same feature that are continuously seeing the similar observation in 2018 and 2021. And then she also concluded in this figure, the shelf input, especially Siberian shelf and Chukchi shelf influence will be very significant in the Arctic, or like at least very strong flux by the flux from the shelf flux indicated by the radium isotopes.
Mariko,
So
excuse me for interrupting, but I think you need to come to an end.
Oh, sorry. Okay, so it's important. And then there is many transporter drift experiment and result are showing here. But it's basically the very changing, a lot of changing and occurring in the shelf area. Shelf area and transporter drift is important to monitor. And also recent experiment, recent result for Nishino et al. Showing the strong low oxygen water transported to the northern part of Chukchi borderland, which is most likely a higher trace metal concentration, which is changing significantly significantly for the trace metal interaction within the water column. And also iron might be oxidized quickly and created the iron hydroxide, which is another source to be actually scavenging rest of the particle, rest of the other trace metal as a scavenging factor. But anyway, it is important to measuring a trace metal, but it was difficult. So it's like, what are you gonna do? And then maybe I was too slow to speak, so I'm not gonna say too much detail, but it's like what I do recently is focusing on the trace metal and nutrient to measuring shipboard analysis. But it's focusing on the silicate because silicate was polymerized with the frozen samples, which is difficult to do some of the people to measuring on the ship. So the current measurement is this system to do, but if you don't have access to it, cannot do. And also this system interfere for matrix effect by the salinity. So sample, if you want to measure seawater or sea ice at the same time, it's difficult. So this system I have developed on doesn't have a matrix effect. And also on this system can do for automatically diluted with the pump to be a series of dilution to make a standard curve automatically. So you don't need to making everything by hand anymore. So then using this system on the boat and compare with the historical system and my system, and looks like a 1-to-1 ratio, so we can use those. And I presented during the ocean science meeting recently to using the same equipment to analyzing aluminum, which is a trace metal, my favorite metals. And aluminum seems to be not much contaminated from sampling system, so we can making a measurement and collecting sample at the same time. And then Jamstec currently building icebreaker, which is also building a trace metal capability laboratory container and sampling equipment, which will be expand for the trace metal capability on the at least for this ship. And there also are measuring, but the shore-based system, setting up the shore-based analysis system at the moment. Sorry about this, it was very slow speaking. This is all I wanted to say today. Thank you.
You're muted, Øyvind.
Yeah, thank you. So you saw me speaking. So thank you very much, Mariko. You had a lot on your heart today, and that's fine. And
I'm so sorry.
A lot of interesting stuff. So I think maybe we should have, if there is anyone having a question, then we should, Ara, go ahead.
Yeah, thank you, Mariko, for a nice presentation.
Hi.
I wondered, with regard to sources of trace metals to the Arctic Ocean, so are the, you mentioned the meteoric water, so I guess also the riverine water might be important, and
Yeah.
as this increased supply of riverine water to the Arctic Ocean, and I guess also these will have higher concentrations of various metals because of the permafrost melt. So
Yeah.
do we anticipate that this will significantly change the trace metal budget in the Arctic Ocean? So it's a huge effect or a relatively small effect? What is the sort of the expectations?
It's we don't know that what is gonna be happening, but it's like there's certainly the shelf at the permafrost, the frozen land, will releasing the nitrogen as well. And then iron is already there. So it's like a nitrogen will be coming around, will going to be impact for the biological activity. Certainly metal will going to increase. That's I expected, one thing. The riverine input, yes, that's also like a transport, but depend on the type of the river, because the Siberian river is more like an organic compound. So it's a lot of organic combined with the metals. So it's together to transport longer. But it's a Mackenzie River, for example, is more lithogenic. So it's like the rock base. The rock base is not really the preserve for iron with as a colloidal phase that much, but so might be a lithogenic phase is more like a scavenging factor to absorb something in the floating in the metal, to absorb into it if it has this release at the same time absorb. So it's like a both, like the increase, but at the same time absorbed, the scavenged and drop. That is we expected. So it's really complicated, depend on the location and the water might be a different. But that's what I can say. Okay. But it's like,
Thank you.
Interesting.
Okay, thank you so much.
So we have one more question, Manuel.
Yes, one quick question. Thank you, Mariko. Maybe you said and forgot, and I didn't heard you saying about that. But are there any historical data on trace metal in the Arctic that can be used to verify long-term changes in some concentrations in some areas or not?
Are you asking in the water column, right?
Yeah.
It's metal itself. It was like the first geotrace before geotraces 2007, I think. That's the European group of people were measuring for the metals on the European side. That's like probably the oldest data set. There's some of the river trace metal data set was like a long time history they have, but the water column itself was not much. That's at least my understanding. That they might have a data, but it's very difficult to evaluate was that contaminated or not contaminated, especially very low version in deep water. Maybe coastal or estuary was a high value. And also in a Chakchi Shelf area, we have some of the data for old like a 20 years old data set, which is very high for iron. So it's probably not contamination, but 20 nanomolar or 10 nanomolar. But it's open ocean is 1 nanomolar or less.
All right, thank you very much, Mariko. And I agree with you that Toyama is a very beautiful city. I've been as fortunate to go there and also have some of the excellent seafood in Toyama.
Hopefully we can do it.
Maybe we can go and have a seafood together one time. So the third and final speaker of today is Mats Reinholdt Jensen from the University of Tromsø. You have the title Biological Monitoring of Vertebrates in Remote Arctic Regions Using Environmental DNA. And are you ready?
Yes, I'm ready.
That's perfect. And the screen is yours.
Just share my screen. All right, are you seeing this?
Yes, we are. Perfect.
Yes, perfect. All right, so hello everyone. My name is Mats Reinholdt Jensen. It's actually my first Synoptic Arctic Survey meeting. So I'll also just happy to meet people who are interested in the Arctic because that's one of my interests at the moment. So I just thought I would start with a quick bio of myself, and then I will present a specific study later on. But I'm a biologist and I work primarily with environmental DNA in aquatic ecosystems. And I did both my PhD and my postdoc at Aarhus University in Denmark. But I have now moved, starting 1st of December, to Tromsø, so working at UIT here in Norway. And like I said, I'm very fascinated by these high Arctic ecosystems, and I'm hoping to do Arctic biomonitoring with eDNA moving forwards. And that's why I think initiatives like this one is so great, because that gives you a chance to get a more complete picture when we're talking biomonitoring in the Arctic. Yes, and I just hope that today I will get to know some new faces and hopefully we can discuss Arctic science together in the future. All right, for my PhD, I worked with 3 different chapters, one being in the open ocean, working specifically with eDNA and whale sharks. And then I did a project in coastal ecosystems focusing on short-term changes in eDNA. And then I've done one focusing on macro invertebrates in small rivers in Denmark. And then for my postdoc, I moved on to work both with insect museomics, so sequencing historic specimens of butterflies from museum collections. And I continued my work with whale sharks, sampling basically globally. But then I also did this one study where the focus was on eDNA gradients and where we had the opportunity to go to Greenland and sample from the south all the way to the beginning of the Northeast National Park. And this is the study I will be talking about today. So the study was published in Diversity and Distributions, and it's titled Distinct Latitudinal Community Patterns of Arctic Marine Vertebrates Along the East Greenlandic Coast Detected by Environmental DNA. And just a quick introduction, I guess most of you know, but Greenland is very remote. It is sparsely populated, and it is highly affected by climate change and pressures from fishing. And fisheries are incredibly important in Greenland. It's actually been estimated that it's more than 90% of the export value coming from fisheries. But this fishing fleet mostly targets areas that are off the shelf, where they target shrimps, Greenland halibut, and redfish in particular. And most of the areas inside the fjords are really not targeted by the fishermen. There might be a few locals that visit these areas and fish there, but not the big fishing fleets. So that means we really have these pristine, large, untouched areas. And the current biomonitoring data that we have from these areas is based on sporadic research cruises that have gone there from time to time. But of course, it's difficult to monitor these areas given its remoteness and inaccessibility. So I'm working under the assumption that climate change is likely to drive range shifts and alter suitable habitats for the species that live in these areas. And therefore, I'm interested in whether we can use eDNA to infer species distributions and community compositions. And I'm very interested in, at least moving forwards, looking further into whether the fish or vertebrate communities are in fact moving northwards. I should say that this study was largely exploratory and opportunistic. I was just asked as a kind of wildcard if I wanted to go to Greenland, and I said yes immediately. But we did go on this trip with Knud Rasmussen, which is one of the SAS registered cruises. All right, so for the sampling, we started down here in the south, where I hope you can see this is station 1, station 2. And then we basically had this 2,400 kilometer waterway distance all the way up to the northeast Greenland here. I should also start off by saying that we had far from perfect sampling conditions. If you're used to working with eDNA, you know that you should be aware of contamination issues as well. And I had to share my Niskin bottle with people who didn't care much for wearing gloves and so on. And then I also had some inconsistent sampling in the sense that I couldn't get water from the depths that I particularly wanted because I was low in the priority list. But keeping that in mind, we had 29 stations sampled, where at each station we took 2 surface samples and 2 deep samples. And then I split the stations into southern Greenland and southeastern Greenland, and then northeast Greenland. And alongside the eDNA samples, we also collected CTD measurements, so temperature and salinity. Oh yeah, I should also say that in this picture down here, you can see how the water is filtered through this tiny Sterivex filter. And this is what retains the DNA that we then sequence later on. This is a terrible plot, but it's just to illustrate my issues. So if you look here at station 1, you can see that we took a surface sample here, and then we basically took a pelagic sample here, and this would be the depth at the station. And what I really wanted was to take a sample that was just above the seabed, but in many instances, also here, for example, it was actually a pelagic sample rather than a bottom sample because they didn't want to go so deep with the CTD. So this is what I mean when I talk about some issues. Anyway, what we went on with was we, in parallel with this eDNA sampling, also sequenced some 12S regions of 87 Greenlandic fish species. And these are primarily from the Zoological Museum in Copenhagen. And out of these 87 Greenland Greenlandic fish species, 42 of them were previously missing in our reference databases. So this is to give ourselves a better chance at actually identifying species from the water samples. Then we obviously need to have a reference database that we can use for identifying. And we had a particular focus on eelpouts here, so the genus Lycodes, and other non-commercial species, because these are oftentimes the species that aren't in the databases because they have no huge commercial value. And then we did eDNA metabarcoding of the water samples, and we did run into some issues. So we ended up pooling the data from the 2 samples we took in the surface and from the 2 samples we took in the deeper waters. And we used a primer set called TELE-02 and one called ELAS-02. And basically, they're meant to target teleosts and elasmobranches, but in reality, they target fishes and other vertebrates. So you're also going to get whales and birds and so on. And then we had it sequenced by the company Novogene on a NovaSeq sequencer. And I know that not everyone here is a genetics specialist, so I just included this as well. This is just to show you that when we talk about DNA barcoding, we usually just have a single individual, a single specimen that we're trying to amplify this particular genetic region for, and we end up with a sequence for that particular species. But when we're talking metabarcoding, we start with a sample that is a complex sample consisting of DNA from many species at the same time. And here the idea is that based off of this complex sample, we will get a list of species that were present in that sample. And since we're using fish primers, it would primarily be fish that we hope to see on this other end. Okay, so I've just got all the results listed here by text, and then I'll walk you through some figures afterwards. But basically, with the barcodes that we generated, we managed to get the percentage of coverage of these species that are known to exist in Greenland up to 87%, and it was 74% previously. So we really did improve our odds for identifying species in the water masses. Like I said, we had some issues regarding this human contamination in the eDNA samples. It's a bit annoying, but it's something you just got to remove, and then hopefully the rest of the data is still sufficient to get these patterns out of the data that you're interested in. We generally found very well matching patterns between what we found with eDNA and what we know about where the species are supposed to be. But keep in mind, there are many unknown distributions. So fishes that have only been caught a few times, or maybe fishes from the very deep, you wouldn't necessarily know what their distributions are off of a few findings. So we did find well-matching patterns, but some of them we obviously don't know with certainty where they are. And then we found this quite interesting turnover in the sequencing read counts when we went from the south and southeast to the northeast samples, where it was hugely dominated by capelin in the south and southeast. And then it was taken over by polar cod when we went to the northeast. So here we're seeing a kind of change on a latitudinal gradient in what species dominates these communities. And we found completely distinct vertebrate communities in the northeast fjords as well. And as expected, we also found a lower taxon richness or species richness in the northeast surface samples. We also did this Permanova test, which showed that about 27% of the variation in the community composition could be explained by the area that we sampled, meaning south, southeast, or northeast, and that 7% could be explained by the depth, meaning surface or deep sample. All right, this is a stacked bar plot, and all I want to do here is to illustrate what I just said about this transition from Melotus villosus. So the capelin is the orange one, dominating generally in the south and southeast areas, and then it's taken over by this blue Boyogadus sida, so the polar cod, when we enter the northeast samples. There was also a lot of stuff going on in the deep, but I think this one was the the primary thing that I want to highlight here. And this one is a bit crazy, but I'll walk you through it. So out here on the right, you have all the samples that we collected, and they're named according to the stations. And down here, you have all the species or taxa that we detected in total. And then this is a hierarchical clustering on both ends. So what that means is if two species cluster together here, it means they're often found in the same sample. And if two samples cluster together, as can be seen here, that means they have quite a similar species composition in this sample. And then a dark green will mean that the species was found, and a light green will mean that the species wasn't found. But all I want to show you here is that we found, if you look at this pink color out here, we found a pretty good clustering that the northeast samples generally clustered together, and also being split up in whether it was surface samples or deep samples. And if you look out here, there's a lot of dark green here, and these are basically species that are kind of distinct for the northeast community. And these are species that make a lot of sense. So for example, Arctogadus glacialis, this is a highly ice-dependent species, so it makes sense that we find it the further north we go. And you also have, for example, this Lycurgus polaris, one of these eel pouts I was talking about, which is an Arctic specialist. So in general, it made sense with the species that were kind of unique to this northeast Greenland community. All right, here you have some NMDS plots, and this first plot here, I just want you to see how the northeast sample communities, they kind of they're at least diverging from the south and southeast, which are a bit more mixed. And this one is based on relative abundance, so read counts of the specific species we found, whereas this one on the right is just based on presence absence, so whether the species was found or not. And basically, to me, it's the two same takeaway messages, that the northeast, so the pink one, is what diverges the most when you see the grand scheme of things. And here what you can see is the richness, so the amount of species or taxa detected. And like I said, we, in the surface waters at least, found generally fewer species for each sample we took in the northeast compared to the south and southeast. And although it appears to be the case as well in the deep samples, this was not statistically significant. And we generally found some species that were only found in either the south, the southeast, or the northeast. And these made a lot of sense in terms of what we know about their whereabouts, their distributions, basically. All right, so for the discussion, it was easy to get rid of this contamination issue that we had, because the only contamination was human, but it does really annoy you when you have to remove a large fraction of your sequencing data just because it's human DNA. We did find distinct communities in the northeast fjords, but we have to speculate whether it is in fact a fjord signal or whether it's a signal of going up to higher latitude. Because I don't know if you recall from the map I showed, but basically in the south and southeast, we were taking samples at the mouth of the fjords, whereas in the northeast, we're basically going all the way into Kaiser Franz Josef Fjord and sampling in a more sheltered area. So it's kind of hard for us to differentiate between a fjord signal and a latitudinal signal in this study. We didn't find any super controversial species at all, and I should say luckily, because this actually means that our method is really useful for pinpointing what we expect to be near exact distribution patterns of these species. The most controversial we found was that herring and salmon were maybe a bit further north than expected. These are also very common food items, so there might be a question of whether this could be contamination, but it wasn't much further north than what has been reported already. And then we found poor beagle, which is a shark species in one sample in southern Greenland. But this is actually a species that has been reported by fishermen in Greenland in the last couple of years. And they are actually speculating that this could be a real case of a species that has come with climate change because it wasn't there if we're talking 10, 20 years ago. We also had a slight issue with one species, a short-tailed skate, and here I'm writing Arctic skate, maybe, and it's because the taxonomists still don't agree whether this is in fact one or two species. And if it was the short-tailed skate, we found it further north than it should be, but if they are in fact the same species, then it would make sense. So these are the most controversial findings we had, and to me, this is not controversial at all. We had one funny observation, and this is very speculative for now, but every time we found a lot of DNA from ringed seal, we also found a lot of DNA from polar cod. And we speculate that this could be a case of seeing trophic interactions from eDNA. And why we're saying that is because the ringed seal's favorite food item is the polar cod. So it would make sense to us that it's actively searching for its food. We had some issues in this study with some confounding factors. We have seasonality and the sheltered versus unsheltered areas where we sampled either in the mouth or inside the fjord, which kind of makes firm conclusions tough to make. But regarding my initial question, whether species are moving northwards, we basically need a temporal study design to tease apart whether any of this northward movement is a response to climate change or other anthropogenic impacts, or whether they could in fact just be species that have always been there, but we just haven't seen them before. Remember, these areas aren't well monitored to begin with, so that could also easily be the case. And that's why I advocate for obtaining such baseline data on biodiversity. It's important to establish where the species are in order to understand where they're moving with climate change. But I think the main message from me today is that this tool works, and I would definitely recommend implementing it in future monitoring of the Greenland vertebrate fauna, and especially in light of global change. I have one take-home message for you today, and it's an easy one to remember. Go to Greenland, it's amazing. I really enjoyed my trip there, and it's so beautiful. So I'm just gonna end with showing you some pictures from Greenland. And with that, I just have one slide left, and this is because I have in fact moved to Tromsø, like I said. And now I'm hoping to expand on my Arctic biomonitoring. So what you see here, these are all the yellow stations that I've just been talking about. I've also been part of a study in Ilulissat in Western Greenland. But what I'm hoping to do now is to get data from two cruises, one going into the Nares Strait and into the Lincoln Sea, and one that will basically do a transect getting close to the North Pole. This is what I hope to get data for in the coming year or so, but it's still a bit uncertain what kind of data I will get out of this, but it's definitely my plan to work with this moving forwards. And with that, I will take questions if you have any.
Thank you, Mats, for a very interesting presentation. I think we should just open up for questions to Mats while we're waiting for someone to raise their hands. Maybe I can just ask, oh, Ari is there already. Ara, go ahead. You're muted now, I think. You need to turn on your...
Yeah, I need to turn down my mic.
Turn down
Sorry
your
for
mic.
that, okay. Thank you so much, Mats. That was quite interesting. Well, you answered some of my questions during your presentation. I was just wondering more about what could cause this gradient, the latitudinal gradient in species. I mean, the water moss is mostly the same, right? You are in the East Greenland current, so you expect a continuum of species, I would guess. So have you reflected more on what can cause latitudinal gradients in the species richness? And also my second question would be about, I mean, this eDNA, it flows with the water, right? So is there any, how long can it sort of stay in the water, and is the risk that eDNA can be produced in one region and then flow to another region where you measure it? So how, you know, how firmly do you identify the habitat or the location of the species? Thank you.
Yes, those are two very good and very important questions when we're talking eDNA. Obviously, the properties of the water masses matter in terms of what species can inhabit those waters. I have to say that I don't know so much about the actual water mass properties, but what we did was we measured with CTDs along the way, and we could see that it definitely was a lot cooler when we went inside the fjords. So the water profiles were quite different inside the fjords. But you're right that on the kind of coast of Greenland, you would probably expect that a lot of the species can in fact live on the entire stretch because it's very similar water masses. Regarding your second one, there is a lot of research going on with regards to kind of the spread of eDNA in water. But my general impression, and I think many others, is that it's a very local signal you get, because when DNA is released into the environment, be it a fish or whatever that releases it, it would very quickly become so diluted that you do not detect it if it's just following the water wherever the water is going. So we, at least based on research in Denmark, very often only get the species you would expect from you know, your very local surroundings, and it would be rare to have input from, you know, the deep sea if you're sampling the coastal area and such.
Thank you.
And so just a quick follow-up on what's going from presence to concentration. Is it possible with any DNA technique? Or?
So that's another hot topic. There are a lot of fisheries that are interested in whether we can use this eDNA technique to inform ourselves about where the stocks actually are, because that would make their life easier. But I would say that for now, I'm fairly confident in the presence-absence strategy. But when we're moving into let's say the read counts that I'm working with here, we talk about what's called relative abundances. So how often do you find a sequence matching one species compared to another? And what I would use that for is to kind of infer what are the dominant taxa in my sample. But there are also many biases associated with this. So one thing is PCR bias. If one species is amplified over another, then your final output is not going to reflect what your sample actually was in the beginning. But if we're talking actual concentrations, then I think it would be better to use another approach, which is qPCR, where you basically get a amount of copies per volume water in your sample, and the question is then, how do you translate that into, say, biomass or something like that? And it could be that one species of fish just pooped right where you took your sample, and then it reflects one individual quite heavily, but it could also be that it's, you know, a whole school of fish just swam by and they all deposited a little bit. So these are very important questions in eDNA research in general, but it's something that a lot of effort is being put into answering these days.
All right, excellent. Good to know that there are scientific questions of importance ahead of us into the future that we also need to answer, I guess. All right, thank you to all the speakers, the excellent presentations. I enjoyed them very much. And thank everyone else for coming and attending, and look forward, I hope, to see you next time, which is on the 22nd of April, same time, where we will have a new SAS seminar. Information about the SAS seminar can be found on our Synoptic Arctic Survey webpage. Thank you very much, and have a good day, good night, depending on where you are in the time zone. Bye-bye.